All right, this is Ms. Chang here, bringing to you session 5, week 5-4 to 5-10, and then this week's topic is on STR profiling. So earlier in the year, or I guess the last 2 weeks, we've been talking about DNA profiling, and then two different forms of DNA profiling. You have RFLP, restriction fragment length polymorphisms, which analyzes VNTR, variable number tandem repeats, and then you also were introduced to STRs, which is the current method of DNA profiling, which stands for short tandem repeats. Hopefully, before you watch this session, that you watch the Bozeman video that I posted, so that you are a little more aware about what's going on. Our objectives for this week is that students should be able to support an argument whether it's a coincidence that two individuals share DNA by analyzing the probability of having a matching partial profile and using supporting evidence. And second objective is students will be able to support an argument whether DNA found at an earthquake site may belong to a missing man or his cousin by comparing STR profiles between two unknown samples in the missing man's biological parents. So we have 2 graded assignments this week. They are posted on Google Classroom. The first one is the document labeled Mixed Up Brothers of Bogota. It is 5 points, all tasks. And then we're going to have live tutorials on this worksheet during office hours this Tuesday because MCPS as well as administration has asked that we get more face-to-face participation up. So we're going to use office hours just more than just a check-in at this point, so that we can walk you through the problems and then you don't have anything to worry about afterwards. The second assignment is earthquake victims. It is also 5 points, all tasks of these 2 are completion, and these office hours will be this Thursday on the 7th. All right, so today's activity that you're going to be the walkthrough for this video is the document titled Forensics DNA Profiling Using STR. So this is a tutorial on how to do this worksheet so that you could do the 2 graded assignments. So hopefully you watched the Bozeman video. If you have not, please go ahead and watch that first. But we're going to go introduce you to the basics of DNA profiling. So you could do the assignment that you see on my screen right now while we're walking through this. It's not graded, but I do appreciate that you do it so that you understand how to do our 2 graded assignments this week. So part 1, DNA profiling basics. While most of the genome is identical among individuals of the same species, differences do exist. DNA profiles takes advantage of these differences. DNA profiling uses a category of DNA variations called short tandem repeats. So you do need to remember what STRs or short tandem repeats are. So STRs are comprised of units of bases. So bases is how we measure the length of DNA fragments for those who do not recall or less than 2 weeks ago on DNA. It's usually 2 to 5 bases long and repeat multiple times. So STRs, just to summarize it, STRs are 2 to 5 base pairs repeats, just like VNTRs. And they're found throughout our human genomes, and they're pretty unique because of the fact that they do not code for protein, that there are a lot of mutations here, and then when it's passed on from generation to generation, it is not repaired because mutations don't really matter to your body because it's not coding for anything. So there's a lot of variations in these regions. So they're usually 2 to 5 base pairs long, going back to the sentence, and they repeat multiple times. The repeat units are found in different locations or loci throughout the genome. So the word loci is very important here. So each STR has multiple alleles or variants, each defined by the number of regions units or by the length of the sequence. They are surrounded by non-repeating segments of DNA known as flanking regions. For example, the STR allele in figure 1 could be designated as 6 because the repeat unit, GATA, is repeated 6 times, or as 70 base pairs, which BP stands for base pairs because it is 70 BP in length, including the flanking regions. A different allele of the same STR would have a different number of GATA repeat units with the same flanking regions. Flanking regions are important because knowing their sequence enables geneticists to isolate the STRs using polymerase chain reaction, or PCR amplification. So just looking at this image here, the flanking regions are going to be at the beginning as well as the end of the repeat sequence. So the word flanking in English means that it's on the outsides or on either side of it. And it's also nonsense DNA, but you don't see the repeat. So figure 1, each of the rectangles represents a repeat unit. So you see GATA is our repeating unit for this particular loci. And this loci, let me look at what they called this gene earlier. So let me go back. Oh, that's not it. So for this particular loci, they don't even mention it. So for this particular loci, the repeat unit is GATA. So once you see it say GATA, you want to see how many times it says GATA, and that tells you the number of repeats. So I look through the sequence and I see GATA starts right here. So this is the first repeat. Then the second one is GATA, 3, 4, 5, 6. So there's 6 repeats here. So there's a 6 repeat sequence, as we mentioned before. So with many repeats, writing the sequence can be very unwieldy. So geneticists use shorthand. So what you see is the traditional way of writing it out fully, and it's quite long. It's 70 base pairs long. So unless you want to write this GATA symbol all the way out, we use shorthand. So shorthand looks like this. So the repeat unit, which is GATA here, GATA is put in brackets, and then we put the number of repeats outside. So since this repeats 1, 2, 3, 4, 5, 6 times, we say GATA in brackets and put a little 6 there. All right, so using the theories from the introduction, we're going to go ahead and analyze this little STR fragment. So question number 1 asks to identify the flanking sequences and the number of repeat units GAAT in the following STR known as TPOX in human chromosome 2. So essentially what we want to do is skip all the nonsense and go straight to GAA2. So what you want to do is scan everything until you find the first GAAT. So I'm going to look starting from the left to the right. I'm going to look for a G first, and then if the G is not preceded by AAT, keep on looking. It doesn't matter if it's close, and I see my first GAAT here. So as long as it's followed by a subsequent GAAT, you have a repeating sequence. So it looks like my first flanking sequence, anything that's not that GAAT repeating pattern, is going to be our flanking sequence. So the first one is CCACAGGTAAT. You could do it capital or not, it doesn't really matter. But this is our first flanking sequence, and we care about this because it changes the size of our STR fragments. It's also where our primers would bind for PCR so that we can replicate the fragments. So after it first repeated, I'm just going to go ahead and start counting them. So after the first GAAT, I'm going to put a little dash between each one and see how many times I see GAAT, and I'm going to go ahead and count them. So it looks like I have the last GAAT there, and I'm just going to go ahead and count them. 1, 2, 3, 4, 5, and then everything after that will be your end flanking sequence. So GCCT, not GAAT. So I'm going to go ahead and type that up as my other flanking sequence. GCCTAAGTGCC. So those are my flanking sequences. And then the number of repeat units. So what I just counted up there, how many times you see GAAT repeated in tandem. So back to back to back. So we saw it was 5 different repeats. So when you express this as shorthand in number 2, what you want to do is just open up your bracket with the square brackets, write the repeat sequence for TPOX, GAAT, and then in subscript, what you want to write is 5 for the number of repeats down here. And let me make it subscript, and that is how you express this in shorthand. So here, STR is inherited just like any gene or segment of DNA. Every individual has 2 alleles per STR, one inherited from each parent. So that is a key idea that you're going to see time and time again. So everybody has 2 alleles, just like your hair color or blood type. You need one from mom, one from dad, 50% of your DNA from each parent to give you your particular STR genotype. However, many different alleles are often present within the population. If the inherited alleles for a given STR in an individual are identical, in other words, contain the same number of repeat units, that individual is homozygous for that STR. If the individual has inherited two different alleles for a given STR, he or she is heterozygous for that STR. Figure 2 shows a simple model of STR inheritance. So let's look at the model of inheritance for a single STR locus. So the number of repeats, the numbers refer to the allele number. So here, the mother is homozygous for an allele with 10 repeat units. That means from the mom, that from grandmother and grandfather on her side, so her mom and dad, each gave her a 10 repeat unit. So for the given locus, so if we're saying TPOX, that you see GAAT repeated 10 times for her first allele, but also her second allele also has GAAT 10 times on her second allele. So that is a homozygous genotype. However, dad here is 1214. What that means is that dad at the same locus, STR locus, has the allele number 12, which means that for this given locus, presuming it's TPOX, also has the sequence GAAT, but instead of 10 repeats, it's a 12 repeat for the first allele, either from mom or dad, and then the 14 represents that the sequence GAAT repeated itself 14 times. So what that means is that you have to draw a Punnett square. This is going to be very important now, where you want to see what mom and dad can give them. So you want to draw a Punnett square. And then mom only has two alleles. Remember, you put one allele on each one. So both of her alleles are 10. And then dad, though, is 12 and 14. So 100% of mom's kids will get the 10 alleles. So I put a 10 for each of the possible four squares. But the dad has 12 and 14. So the first half, so the top two rows, will have a 12. And in the bottom two rows, will have a 14. So these are the two possible genotypes. You get the 10-12 repeat as well as the 10-14 repeat for their offspring. And it looks like the daughter has the 10-12 repeat. So it's possible a brother will also have 10-12, or they could also have 10-14. It's going to be a 50-50% chance. So the father is heterozygous in this STR locus with one allele having 12 repeats and the other is 14. The daughter is heterozygous, two different locus, two different alleles, two different alleles having inherited 10 from the mother and 12 from the father. So this is the daughter's genotype. Okay, so the question here is there any way the daughter in figure 2, our last figure, could have been homozygous for the STR shown? Explain your answer. So the only way that the offspring could have been homozygous is that if mother is 10-10, father will also have to have at least 110 for a homozygous offspring. So the answer here is going to be no. No. The daughter cannot be homozygous based on the data in figure 2. Both mom and dad have to share at least one allele in common for to pass down to their offspring. All right. So that is our answer there. Because mom and dad don't have a common allele, you cannot have heterozygous offspring. So 4, using the diagram on the right, identify all the possible STR genotypes the son can inherit at this particular locus. So what we want to do is find the possible genotypes the kids have based on mom and dad. So this is something that you're going to have to use for our earthquake victims scenarios. Can it be their son based on the parents' genotype? So let's go ahead and do a Punnett score. So I draw the box, and then I want to make sure that mom is on top. We got 5 and a 6. Father's on the side, 4 and 11. So based on the genotypes, mom can give 50% of her kids a 5, and the other 50% of her kids will get a 6 repeat. So father, like mother, is also heterozygous, so 50% of his kids will have a 4, but the other 50% will have an 11. So based on this cross, we have 4 possible genotypes. We have a 5-4, or I'm going to rearrange it so that the smaller number comes first, a 4-5 genotype. We also have a 6-4 or 4-6 genotype. We also have a 5-11 genotype. And last but not least, 6-11 genotype. So these are the different variations of genotypes that are possible based on this cross. So remember when we talked about probability and permutations? Well, based on the possible alleles that mom and dad have, based on independent selection, we have 4 possible possibilities of 4 possible outcomes of genotypes. Got 4-5, 4-6, 5-11, and 6-11 for this particular cross seen up here. So how is DNA profiling done? So with RFLP, we showed you guys that we used gel electrophoresis and the southern hybridization technique. It's very costly, very time-consuming, and it's just not as precise. So we've actually gone away from it. So even though last week you guys were looking at gels and the bands, that's not very precise anymore. So scientists analyze DNA by first extracting and isolating the other materials in an individual cell. So once extracted, the targeted STRs are amplified by PCR. So this is a little different than RFLP. All of the DNA has to already be there in the RFLP technique. So you need to have a sizable DNA sample, and then they didn't copy it back then. So whatever you had at the crime scene was all you had. And if you didn't have enough blood, you couldn't do the RFLP technique, and it wouldn't show up on a gel. So with STR, though, you could use PCR to amplify it. So I will post a video to show you how PCR amplification is done, but as long as you have one copy, one strand of that DNA, you can make infinite copies with PCR. So different STRs are tagged using a different fluorescent marker so that they can be easily detected and differentiated. For efficiency, many STRs are analyzed at once during genetic profiling. You may be familiar with gel electrophoresis as a technique for separating DNA fragments by size. Researchers analyzing many fragments from many samples use a variation of the technique called capillary electrophoresis, which is faster and easily automated. Figure 3 shows a model of capillary electrophoresis. As DNA samples go through the system, the results appear in an electrophoregram, or a graph showing the quantity of light at specific wavelengths detected over time. DNA fragments appear at different color peaks that can be compared across samples and to the DNA ladder. So it's better explained when you look at the picture. So here's figure 3 here. So a simplified schematic of how an electrophoregram for a single STR is generated. In this example, the individual has one allele with 5 repeats and another with 11 repeats. So here are the 2 different STR strands, the 5 repeat unit and the 11 repeat unit. So during PCR, the DNA fragments are amplified and a fluorescent tag is applied. So amplification is where they copied it, and then they're going to put a tag, which is pretty much a primer is going to bind to that, which is a fluorescent marker that will show up under like blacklight or something. And it will also show up in the computer. So after PCR, the DNA samples are run through ultra-thin capillary tubes filled with a polymer. An electrical field is applied, causing the negatively charged DNA fragments to move towards a positively charged electrode with larger DNA fragments moving more slowly than smaller fragments. As the sample is drawn through the capillary tube, a laser excites the tags in the emitted fluorescence is measured by an electronic camera, a detector. Fragments of the same length pass the detector at the same size. So running fragments of known DNA sizes, known as DNA ladders or DNA standards, through the system allows the scientists to determine the size of alleles in the DNA samples. The alleles are shown in the electrophorograms as labeled peaks. This technique also allows scientists to construct a graph that relates fragment sizes, measures the number of base pairs versus time. Computer software uses DNA from a detector to determine the size of alleles in the DNA samples. In this example, 55 base pairs, allele 5, and 79 base pairs, allele 11. So essentially what happens is that, essentially what we have on our screen now is a sample DNA profile looking at 4 different loci, so 4 different STR loci. So an electrophoregram for an individual showing the alleles for 4 loci, D8S1179, D2-1S11, D7S820, and CSF1PO. Each locus shows the alleles below its peaks in the electrophoregram. The number of repeats for each allele is indicated. So for number 5, they ask, examine the electrophoregram in Figure 4. Determine how you determine whether an individual is heterozygous or homozygous for a particular STR. So the number of flanking sequences does change the size of your fragments. So our scale is anywhere between 100 and 300 base pairs. And then nicely, because of the fact that each repeat is very small, remember it's anywhere between, they said around 2 to 7 base pairs, that even if you have very different numbers, that the distance between the 2 peaks for a heterozygous genotype would not be very far. So for the first genotype, we have 1316. So let me write 1316 here. So this is our first genotype, 1316 at D8S179. So what that means is that when you see 2 different peaks, one smaller than the other, obviously, that this is a heterozygous genotype. So you have a 13 repeat, and then you have a 16 repeat, and then this is at the particular locus D8S1179. So I don't know the exact sequence of it, but what that means is that the first peak you see, so further to the left, the faster it was, the smaller the fragment, is that you saw a 13 repeat bleep show up on the screen, and then it goes away. And then all of a sudden, a second bleep comes up for your 16 repeat because it's a little longer, so it took a little longer to get through. And these 2 peaks signify a heterozygous profile. So for at D2-1S11, we have 28, 29. So 28, 29, those are your allele numbers. And once again, they tell you how many repeats there are at the second locus, D2-1S11. So unless they're both the same, this is also going to be heterozygous because you also see 2 different peaks signifying 2 different sizes, one with 28 repeats and one with 29. So you can see that the first peak, a little like almost 200, whereas the 29 repeat is a little larger than 200 base pairs. Then all of a sudden we have D7S820. So this third locus, you only see one peak here, and you only see the peak 9. So what happened here? So if you're paying attention carefully in our last video, is that the numbers correspond to the number of repeats. And if 2 different numbers, then you will be heterozygous. Well, if you're homozygous, what happens? Well, that means mom and dad both gave you the same size. So if they're both the same size STR repeats, the copied segment will be exactly the same. So you would not be able to differentiate between mom and dad's copy of this particular allele. So what ends up happening is that it shows up as one peak because mom's copy of this STR allele is the exact same size as dad. So instead of just drawing a 9, you do 9, 9, and this will be homozygous. And then last but not least, we have CSF1PO. We have 1112. 1112, you get 2 different size peaks. One that is a little above 325, maybe let's say 326. And then the second one is another repeat. Let's say it's 329. It's a little longer. So because it's 2 different sizes, we're going to say this one's heterozygous. So let's go ahead and type up our answer. So examining this electrophoregram in Figure 4, heterozygous STR genotypes result in 2 peaks because of DNA fragments of different sizes, homozygous STR genotypes example 9-9 at D7S820 result in one peak because both alleles are the same fragment length. All right, so let me go ahead and get this all on one page. There you go, that's your answers. So if you have a heterozygous STR genotypes, so that's going to be, for example, your D8S1179, which has the genotype 1316, that's going to result in two peaks because your two DNA fragments are different sizes. You got a shorter one and you have a longer one. When it's homozygous, just like this guy, you're going to see only one peak. Right. So next to each STR locus below, identify if the genotype is homozygous or heterozygous. They were all heterozygous except for one of them. Let's look at the one that was not. So D7S820 was the one that was homozygous, and that's this one. So safe to say the rest of them are heterozygous. Heterozygous. It's two different peaks, heterozygous. One peak is homozygous. Which locus has the longest DNA fragments, and how do we know? So DNA fragments are measured in base pairs, as we mentioned in the introduction. So on the left, we have the smallest size fragments, 100, and the right, we have the largest size fragments. So what we want to see, so going back to the question, which locus or which location is it going to be? This first one, D8S1179, is it going to be the second location, which is D21S11? Or is it going to be the third one, D7S820, or is it going to be the last one, CSF1PO? Since we're looking for the largest one, you want the one that's closest to the right. So that means our answer is going to be right here, CSF1PO. So let's go ahead and type that up, CSF190. Then how do we know? Both fragment lengths were the furthest to right of the scale. Right. Oops, can't spell right now. Being a ladder scale as it increases from left to right. So one of the things that you need to be able to do is to just guesstimate what is the size of this DNA fragment. So I'm just going to go back, and you're going to use it just like a ruler, try to guesstimate how big it was. So as I mentioned before, this one looks like about 326. So it's just slightly to the right of 325. Just like you were to use a ruler, anything in between will be a number. Between here and here will be a number between 325, 350. Since this is just one base pair repeat, let's just assume it's a 4 base pair repeat. That maybe instead of 326, it's 4 base pairs longer. So instead of, I'm going to say approximately 326 and 330 base pairs. So those are the largest. Whereas when you go to the far left, we have things a little under 150 and something that's a little larger than 150. So this one looks like it could be 155. This one looks like it could be 148 or something. So these are much smaller than these guys, about half the size. So here, different individuals have unique sets or profiles of DNA alleles. For many forensic analyses, researchers used a core set of 13 STR loci established in '96 by the FBI laboratory. So these core loci, so this is real, these are the exact names of these different loci, is CSF1PO, FGA, TH01, TPOX, VWA, D3S1358, D5S818, D7S820, D8S1179, D13S317, D16S539, D18S51, and D21S11. In addition, a gene marker called AMEL, A-M-E-L, is used to determine an individual's gender and is analyzed along with the 13 STR loci as part of the individual's genetic fingerprint. When all 13 loci are analyzed, the chance of two people, with the exception of identical twins, having the same exact set of alleles for each of the 13 STR loci is extremely low. Some will put it in the neighborhood of 1 in, let's see how big this is. I don't even remember right here. Let's do million, billions, trillions. So 1 in 10 trillion. So in other words, the chance of a random perfect match of all 13 loci is 1 in 10 trillion. Look at that. The number was already down there. All right, so that's it for this video. What we're gonna have you do now is gonna be these two activities. So they're similar, there's nowhere as long. You wanna analyze the STR profiles of that's given to you for our brothers, and then try to see if where they switched at birth. So we're gonna have a tutorial that's live during office hours tomorrow on Tuesday, 5-5. And then the second activity is also all task earthquake victims. We have 2 missing people during an earthquake. We want to see if they could have been the son of 2 worried parents or maybe the cousin. So this is going to be during office hours this Thursday. Thanks for tuning in. Please show up for office hours.